All right, Vipin, do you want to kick us off and introduce yourself?
Sure. Sorry. Hi, I'm Vipin Suri. I lead discovery at Obsidian Therapeutics. And at Obsidian, we're engaged in really bringing precision pharmacology to supercharge T cells for oncology and other applications. I'm a biochemist and pharmacologist by training, spent time at Pfizer, GSK, and Raise Therapeutics before coming to Obsidian.
Hi, Dennis Underwood. So I lead the core technologies at Agenus, which is a company out in Lexington. So we have a number of challenges ahead of us where we're looking for checkpoint modulators, vaccines, and cell therapies in a combination approach to treat immuno-oncology. So I've been in the industry over 30 years, and probably half of it has been in big pharma. Places like Merck and Bristol Myers Squibb. And the last half, which has been actually the most exciting, is in smaller biotech, including my own tech startup.
So hello, everybody. My name is Christine Lowe. I'm currently at Kymera Therapeutics. Kymera is working on heterobifunctional small molecules to treat diseases of various different types. We believe that these heterobifunctional molecules allow us to exploit the ubiquitin proteasome system to be able to degrade protein targets that are involved in disease pathology. This enables us, we think, to be a completely different modality and to access targets that were previously undruggable in a different way that gene therapy and siRNA or ASOs and other types of novel modalities also have done. So my original training, I'm an immunologist, but being many years now in drug discovery and development across large and small companies, I've become more of maybe a general cell biologist, drug discovery, pharmacologist. And so really enjoying my time at Kymera. The largest companies that I've been at include things like Pfizer. Smallest prior to Kymera was Certris, which was acquired by GSK, small and then became large again, but it's probably part and parcel of how the whole biotech and pharma world cycle goes. So,
all right, thanks everyone. So as you guys can tell, we're in good hands for this panel. So a bit more context on the topic. I think we can all appreciate that tools and technologies can be key drivers of discovery and innovation. And I think we're at a moment now where in recent years we've seen the emergence of technologies that really have the potential to revolutionize how drug discovery is done. So I'm thinking of things from single-cell sequencing and CRISPR to things like machine learning and artificial intelligence. So I think as R&D managers, you're always faced with rapid change and technical innovation in a really unpredictable but exciting environment. And so we thought we'd sort of anchor the conversation around how you manage R&D in that environment and make that balance between innovation and maintaining the current development cycle. So I'll kick it off with a few questions up here, and then we'll open it up to the audience to make sure we address everything that's on your mind with the panelists also. But maybe the first question, it'd be great to just hear from all of you. What do you think are some of the emerging technologies right now that really have the potential to change how we do drug discovery?
Would you like me to start?
Sure.
Yeah. You mentioned some of those. I mean, those are the ones, from tools and technologies that are most exciting. Thank you. Is that better? Can you hear me okay? Oh, thank you. So as Jessica just mentioned, single-cell sequencing and CRISPR, machine learning, et cetera. I mean, these are the emerging technologies. For me, we work in cell therapy. My mind is very much focused on the problems that we face, and I'm excited about a lot of these technologies. I think it's a great idea to have a broad toolkit, but some of the things that I'm specifically very excited about are in gene delivery, using genome engineering with CRISPR or other nucleases for site-specific integration, I think has a great potential, not just in research, but also the way these cell therapies are manufactured. So these are some of the things that I'm excited about.
Dennis?
Okay. So it's really a complex area. So for Agenus, we're looking at immuno-oncology. And if you smash two really complicated fields together, that's what it is. And it's very difficult to work your way through it. You know some of the questions and you know some of the answers, but you don't know everything. So it's a real challenge at every step of the way. And I'm old enough to have seen a number of waves of technology that have come through the industry. Computational sciences, structural biology, combinatorial chemistry, high-throughput screening, genomics, the whole 9 yards. And what tends to happen is that it lifts everybody up. So there's a lot of hype at the beginning. You never actually reach the potential or the hope or the sales pitch of the technology, but it does lift everything up. All of the technologies get integrated and they get plugged into the processes for discovery and development. But one of the things that I think is a real challenge going forward, certainly for immuno-oncology, is the need to get durable responses from a very complex set of diseases. And it seems as though combinations are really important. So to me, the ability to actually search across the field very broadly and understand how small molecules, biologics, cell therapies, vaccines all work together is really critically important. The only way we're going to work our way through that, I think, is through machine learning and AI. There's so much information and data, so much complexity, so many things that we don't really understand that's kind of embedded in the data, that I feel that AI will lead us through that. But again, there's a lot of hype, and in practice, over the years, it'll be integrated uniformly through all of the practices that we do.
I think the question of what's the most exciting technology is a difficult one to answer because there are so many different technologies. I mean, it's amazing that we've hit this inflection point where every day something new is coming out, even with existing technologies. That's what the impressive piece is about it. So, you know, antibodies have existed for quite a while, but when you keep reading about how biologics is sort of turning around and coming up with something new, bispecifics, et cetera, I think it's important also that we don't forget about things that are already there maybe and are hitting that next generation or that new iteration. Something that comes up, because I know you were asking about the balance, I think some of it is really keeping in mind the fit for purpose. I think my panelists were mentioning that we all get caught up in the hype and you hear about something new and it seems very exciting and it seems like it's going to solve all your problems. And as you start going down that road, it may not be quite what you needed or thought you needed or wanted to be able to answer the questions of the day. So I think the tough piece that all of us face is picking and choosing, because ideally we'd like to be able to do it all, but cost, resources, just time, brain bandwidth and everything else sort of comes into play. So I think it's really picking the key technologies that's going to help you address your question now and how that's going to really accelerate what you're doing forward. You know, at Kymera, for example, we take even tried and true, again, coming back to my earlier example of proteomics. It's existed for a while. Mass spec has certainly been there, but it's really exciting to see what the new frontier of how mass spec is pushing forward and getting us down to even more depth of protein analysis, more post-translational modifications, for example, that's also enhancing our understanding about how proteins work, how they're modulated and regulated.
Thanks. And Christine, you started to touch on this, but I'm curious because you've all been in such different companies of a range of different sizes. How do you kind of think about striking the balance between investing in new technologies or maybe following the hype or sticking with a sort of more tried and true approach to drug discovery? And I guess a related question, how does that really inform how you build your R&D organization and culture?
Yeah, that's a great question. So again, we work in cell therapy, so unfortunately, there isn't a tried and true, and there's only a couple of products on the market in the last year, so every day is pretty much a new battle. I mean, the key thing, for me at least, and Christine touched on it, is, you know, these are tools to solve problems. That's very important to keep in mind, is to have a broad toolkit, but make sure that your focus is in solving the problems that are holding back. You know, our goal is really to advance our therapies to patients the fastest we can, the best therapies to patients, right? Now, the second part of your question is actually really interesting as well, which is you know, what are the organizational factors that help in deployment of these tools effectively early as problems strike, and that's a cultural question. So we at Obsidian are blessed with a team that is very much interested in new and emerging technologies and applying those to specific problems. And the important thing to keep in mind again is not to let the toolkit become the problem itself, which sometimes it does, right? Because you end up spending so much time on adapting a tool to a problem that the nature of the problem changes. So these are sort of things: is define the problem that you're trying to solve, have a broad toolkit, have a culture where folks are interested, genuinely interested, and encouraged to explore new tools, and then just make sure that the tool doesn't become an end by itself.
Yeah, the cultural aspect is really, really important. And I think one of the things that I look for in companies, for instance, is a top-down approach to recognizing that innovation is the lifeblood of what we do. And you've got to build a team that's free to think and to be creative. But on the other hand, as a manager, you've got to kind of restrain them because you're going to get work done. You can't be off looking in the corners for new things all the time. So it's a real balancing act, and that's probably the biggest thing that I do from a day-to-day basis, is to encourage people to go looking for new ideas and new approaches, but then to put their noses on the goals and the achievements that we have to reach. Otherwise, we're not going to get therapies. So it is a challenge.
Yeah, it's an interesting question, and maybe I could take it also from a slightly different perspective because, again, you'd asked about organization and culture. So the way different types of companies and different sizes of companies, at least the ones that I've been in, sort of handle things very differently. At a large company, some of the biggest pharmas, they, because of the size and the resource and maybe some of the money aspects, can afford to set up a team to kind of park themselves over in a corner and start to explore some of the emerging technologies without necessarily needing to apply it or integrate it immediately into a pipeline or something that they're doing. That, of course, has its own caveats, because the con of that is that I think those groups sometimes are pushing through technologies and they are making the critical go, no-go decisions on those, but those aren't necessarily seen from the side of the pipeline because, a no-go decision doesn't ever get taken up anywhere in the organization. So some of the work that they do is a little bit lost in that testing of the innovation and the technology. The small companies, unfortunately, or differently, don't have quite the same kind of structure or means in order to do that. So by definition, exactly as my co-panelists have mentioned, you're looking for ways of integrating that solution right away as quickly as you can to solve the problem. The tough part about that is that you're constantly making these decisions or justifying, as the case may be, of why you need that technology. What's the proof that you have and the data that tells you that this is the solution? And that's exactly it. Well, if something's new and emerging, there might not be a whole lot of data yet to prove that this is going to be the solution to our problem. So it's that push-pull, a good one, because it's good that we constantly think about where we need that emerging toolkit to be able to help us on a daily basis.
Great. And I want to come back to sort of a specific example of a technology, and I think, Dennis, you started to touch on this earlier, but maybe if we think about AI, it'd be great to hear just how, in your respective companies, to the extent that you are, how you're using it.
We are not. Most of our most of our intelligence is natural.
That's good.
However, it's something that we're very interested in. Well, the reason we're not is because our problems were elsewhere. It's not that AI is not of interest to us. It's just that we had bigger things to deal with that there were other tools for. However, that being said, it's an investment we started making recently. It's something we're very interested in. We have a protein design platform that can really benefit from AI. We are starting to bring in scientists who have experience in data sciences with the goal of at least learning more about it and how to use it as a tool.
So Agenus is really deeply into machine learning and AI, and we see it as as a necessary component of actually moving forward. But there's something you have to do before you get to that point, and that is to wrestle your data, the data and the information you've got, to the ground. You've got to get it organized and straightened up. You've got to train machine learning and AI algorithms based on something which has a high quality set of data attached to it. So that's partly, our interest in Benchling. I'll put in a plug for Benchling, was to be able to actually do that. But there are probably 10 different sources of information that all feed into decisions that are going to change the way we discover drugs, develop them, and how we combine them in the clinic. And the methods that we pull together to that are really important. So one example is, for instance, where we have a cell therapy unit as well. We're interested in the T-cell response, the repertoire, to therapies. Before being given a therapy, after being given a therapy, and longitudinally along a patient's pipeline. That generates a huge amount of data. And what we're looking for are signatures in the sequence that really point us to various function and outcomes. That's a very complicated problem, lots of data, but one we're really interested and excited about continuing.
So at Kymera right now, we're probably just getting started in a little bit of AI, but maybe from a different perspective, all the way at the beginning of our pipeline, per se, of target selection. You can imagine from what I described, we've got technically the world of proteins at our feet if the heterobifunctional molecules work the way we do, so we can degrade any protein, conceptually. The real question then is, how do you decide what focused set of proteins you're going to work on? And in fact, we're trying to look at AI as a means of being able to help us comb through the literature, comb through clinical data, gather an understanding of which types of proteins might be best suited for our particular approach. So that might be one very proximal way that we're looking at AI. From a personal standpoint, and again, having been at different companies in different settings, I think one of the exciting things or applications for AI is really at the complete opposite end of the spectrum of how do we accelerate clinical development by being able to select our patients or stratify patient responses in a much better mode. Humans, even animals, are very complex systems, so everything that you can boil down to in a minimalist way in a cell or a cell experiment completely changes when you go to the organism. And we are faced with complex diseases that we lump together in one pile, diabetes or lupus or something else, when there's probably the good likelihood that there are subsets within each of those major disease areas. And if somehow we could pinpoint that, it might enable all drug companies, biotechs everywhere, to be able to do a better job of making our clinical trials and development and design much more efficient and effective in the long run to get those therapies out to patients sooner, and the right patients sooner.
Great, thanks. Yeah, I really appreciate that point. As a geneticist, I love this idea of patient stratification and really understanding detail, sort of patient populations at the detailed level. You started to touch on target selection, and I think targets is always a really interesting topic in biotech. And so I'm curious, are there ways that new technologies are helping you go after targets that you thought were previously undruggable? And are there targets you're excited about now that you wouldn't have thought about going after before?
Right. So there are for Obsidian, again, because of the nature of what we do, which is cell therapy, where that's mostly a hammer. Not so much. But if you look at some of the other emerging technologies, I mean, Christine has the perfect example, right? Which is just amazing, which lets you do things that you always wanted to do, but you couldn't because there was no way to inhibit proteins that don't have enzymatic activities or degrade them in a way. So they're certainly opening up a newer space, as are other technologies, CRISPR, et cetera, are opening up new avenues that were not possible before. So there is certainly a greater universe of targets you can go after with the newer modalities.
Yeah, choosing the right target is really the holy grail. And, you know, there's a lot of Me Too drugs out there, which is a good thing. I come from, you know, a background of small molecules. And essentially what you do is iterate your way to a really terrific drug. And Merck used to do that all the time. Pfizer does it. Everybody does it. They're all Me Too's. And the same is true for checkpoint modulators. But, you know, to be able to get into an area where you're seeing a landscape where there are brand new molecular targets is really exciting. And I think one of the things that has turned us on over the last few years is the idea of going directly for the peptides that are presented by MHC molecules. And these are the things that are actually recognized by the immune system. They're the targets actually that you're going after, either with cell therapy where you try to elicit a T cell, educating T cells basically with a vaccine. So we've spent a lot of effort looking at a variety of different tissues, disease tissues, normal tissue, mouse, human, alien, I think, in some cases. And we've been mapping out the phosphopeptidome, the presented ligands, which are post-translationally modified. And these are uniquely interesting because they represent as a signature of what's happening inside the cell. So if the cell is transformed or the cell is infected, then that signature actually tells you a lot about how happy the cell is. So these are all brand new molecular targets. These are targets for cell therapy, either T cell receptor focused approaches, or even if you can do something like create TCR-like antibodies, all of that area is a brand new field, and we've cataloged many of the targets of interest there. So it's really exciting.
Yeah, so I think one other way that I think about also is not only the very exciting, catalog of new targets that are being discovered and identified and hopefully validated every day. It's also, again, and coming back to everything else old is new again. Sometimes targets that people have been chasing for a long time, in fact, may have made drugs for already, but aren't drugged well or perfectly, maybe would be the way to say it. So, like, for example, Kymera is going after a couple of targets that are out there in the field. So yes, you could say that maybe we're Me Too, but thinking about it in a different light, maybe that enzymatic inhibitor isn't working well enough because the protein has more than just one enzymatic function, and you actually need to block multiple aspects of it in order to shut down the disease pathology. So it's coming at the same target, but in a completely different way. It even goes to the aspect of even manufacturing and cell therapies. For example, Synlogic was here, I know I was talking before, where, again, maybe the target or the underlying disease pathology is known, but there hasn't been quite the best way of getting at it. So coming at it with a different aspect of even being able to manufacture a new and better drug by cell therapy, by engineered bacteria and otherwise, might enable us to be able to target already known targets better than we did before.
Great. Well, I want to make sure we save time to open it up to the audience to allow you guys to come in with some questions. So that, I think, if there are questions people have, we'd be happy to take them. I think we're kind of mic limited, right?
So I've got a question. We talked a lot about technologies that might be really hyped, disruptive technologies that might be hyped up. I'm curious from each of the panelists, what technology do you think is actually underhyped and deserves more attention? One technology.
Oh, I don't know. The overhype is a lot easier. I mean, well, I can tell you the problems I face, right? And I'm sure there's solutions out there, but they're underhyped because I don't know how to solve, right? And, a lot of it are very simple problems, like data visualization that you're working on, right? We run a lot of assays. We try to run them the best we can. And there's a lot of data in there, and I feel like we're not able to mine, and we actually use your platform. There's tools out there that can help us do that and not cost, crazy amounts of money. That'll be important. The other thing that's also, I'd be very interested, maybe there isn't a technology, but I'd be very interested in, is, when companies like ours start, we do a lot of new things, we develop a lot of new assays, and not all of them are, done with the precision of a high throughput screen where you have a very specific, assay development path. And it's very hard to sort of combine those data sets to get information. The information is there, but because it's sort of non-standard formats, you got to fill in the blanks on behaviors in the assays. So that can be modeled, and I'm sure there's ways out there, software out there that can model that and tell me that even a collection of non-standard assays, what is it telling us? So either these tools are underhyped or they don't exist, but I can't imagine they're not doable. I mean, you guys do all sorts of wonderful things with Benchling and other software and the AI hype. I mean, this is a simple AI problem, right? I think.
Sounds like it. Saji, you should do it. So that's a really difficult question, actually, because a couple of things spring to mind, but I think the thing that, to me, is really important is the idea that you could understand the signaling mechanisms and the interactions between antigen presenting cell and a T cell, for instance. You can sort of understand it by deduction. So you can do some high-level genetics, etc., and you can knock things out and knock things in and play with certain side chains, etc. But the really important thing to all of that is to understand it from a assembly point of view. And I'm thinking here of cryo-EM and the interaction between an almost atomic resolution method and immunology and oncology, and the ability for receptors to cluster up in membranes and to create their macromolecular assemblies, which are really the signaling and the really effective pieces of the immune system. We all tend to approach every one of our problems like an antibody in a singular kind of a way, or a T-cell receptor. We think of it as a single entity, but it's actually a cluster of things on a cell membrane, which is the signaling thing. So I think that's underhyped. I think there is a huge potential for understanding that and all the ancillary technologies that would go in with it. So that's what comes to mind.
And I think about maybe what's underhyped is maybe some of the older technologies or methodologies that have been sort of left behind, or that people use but isn't sort of first and foremost. So, we're all in the next generation sequencing level where you can get RNA-seq and millions of reads, but at the end of the day, there are still things like old-fashioned PCR, multiplex PCR, where you're looking at 10 genes instead of millions. I think the complexity there is exactly, as my co-panelists again had mentioned, is how do you integrate that information? So it's great that I have 10 genes, I'm looking at 2 cytokines, let's say, and a couple of signaling molecules without querying the whole phosphoproteome. I think there could be very rich understanding in that sort of small data set, if you will, but we haven't mined it to its fullest potential because the data comes in different forms and different formats, and if you're looking at the human eye and human brain to kind of pick out trends, of course we have all our own biases as well. So I think it'd be really nice to be able to figure out even how to use the existing information that we have to do a better job of trying to mine that for the interesting aspects that could help drug discovery and development.
Other questions in the audience?
So a lot of this is looking forward as to how new technologies can influence what we're doing today, but how can the knowledge that we're gaining now sort of make us look back at some of the things that we've been relying on and assuming work? Something that comes to mind is like mouse models, just like something that is so critical to our industry, but you don't hear a lot of innovation going on in something like that. What are some of those older technologies that are being taken for granted and you think we could benefit from revisiting those?
Yeah, I think you're exactly right, and maybe you're sort of following on what I was mentioning sort of in the last bit there. I think there are innovations happening there. There are new mouse models being created and made, and they're not hyped. You don't hear about it unless you start digging deep and you're in that field and you happen to notice that somebody's come out with a new knockout or a gem model of some kind. So I do think there's aspects of it of just keeping your eye on what's already built, and maybe, again, that version 2.0 of whatever exists out there could be, in fact, transformative in its own way. I do think that there's opportunities, again, for thinking about, and that's something we didn't talk about, so a technology that I heard a lot about years ago but seems to have sort of gone into the ether a little bit is organs on a chip, for example. How do you model something that is complex down to something that's almost a minimalist version of it to increase throughput or anything else? So I think that maybe it's just because it's underhyped, but it's worthwhile continuing to monitor some of those changes and developments that are going on because, again, they could be very transformative for some of the aspects of drug discovery and development.
Yeah, it's a good question. I mean, the basic toolkit, some aspects have not changed for a while. But the specific example you give of mouse knockouts, that's an interesting one, right? If you go back 15-ish years, 10, 15 years, there was a company called Lexicon Genetics, and I think there were others too that were just cataloging, making all sorts of knockouts for every gene. But that technology, and then Lexicon sort of went into therapeutics, and I'm not sure where things are. But the knockout technology itself is changing now with CRISPR, conditional knockouts, and all sorts of genome engineering innovations. Maybe the reason we're not hearing about it is there's probably not a systematic effort to do it on a scale that some of the older. But I think it's very valuable. Also, humanized mice models, right, for immunology are becoming more prevalent. But you bring up a good point. Maybe perhaps that's underhyped, right? I mean, that's, yeah. Thanks for the question. It's a good one.
Yeah, it is a good question. But we do a lot of incremental innovation. So given a technology, right, in the background, it gets improved continuously, like transfection, viral transfection of cells, etc. That's a continuous process because although you can do it, it's not being done very well. And there needs to be a shift in that. And people are working on it. So that's, yeah, there's all the hype stuff, but there is a background of a lot of creative work.
I actually have two questions. The first question is about the regulatory hurdles or challenges that you foresee, especially as you bring like breakthrough therapies into the market. And the second question is, how much of your organizational design is built around addressing the problems of your science versus addressing the possible like development challenges and regulatory hurdles that you might need to go to market fast?
Yeah, so I think in our company, I can say right now that we're all focused on the science. It's not as though we completely ignore the regulatory aspects of things, but maybe it's because also specifically Kymera is still within small molecule realm. Slightly larger size small molecules, but still small molecules. So we do feel like there's a well-precedented path, at least for some of these therapies, which is maybe also why we focus there, because we have to prove the point first, that the modality works the way we want it to work from an efficacy and safety standpoint and all the rest of it before we even face the regulatory side of things. It's a great question, though, in terms of how do you, again, sort of balance those aspects of what we do on a daily basis. So I'll let my panelists also provide some other insights.
Yeah, so I think the other dimension here is speed of doing things. And really, the ability for you to discover things and to put them into development is not the end of the job, right? So you need to then get it through an IND, and you got to make it for clinical, etc. So I know Agenus very much sees that it's the whole path, which is really important. And so we spent a lot of time in speeding up the processes for development and manufacture for clinical. And we're in the clinic with our own checkpoint modulators, and we make them, and we've done it incredibly rapidly, but it's primarily because we thought that was really important, and we put a lot of emphasis on it and a lot of resources behind it. So I mean, whoever would start a biotech company if they were a VC? I mean, it's just an impossible task. There's so many challenges all the way through. But the rewards are phenomenal. And keeping that balance, I think, is one of the challenges of managing a biotech well. It's not just discovery. You've got to develop, and you've got to get it out there, and you've got to commercialize it. So how are you going to do that? That's a huge challenge.
Yeah, so again, it's a good question. And you asked specifically about regulatory, which is FDA interactions, and is a relatively rigid process. For cell therapy, that is a huge challenge or opportunity, depending on how you look at it, because there's no precedent. There's very little precedent there. And every step of the way, from the second you say, I'm going to manufacture a cell therapy that I'm going to put in a patient, companies are still, and FDA are still trying to figure out what should the release criteria be, so those details are just emerging. As a small biotech, it's not our primary battle, because this landscape it's going to take a lot of major players to work with FDA and figure out what that needs to look like, and FDA has to release guidance documents, which they've started to, but it's not quite there. The second part of your question, in terms of our organization, again, for Obsidian, we are in the space of regulated cell therapy. Our core value that we bring is this technology for regulation, so we focus on the early science. The regulatory questions still come up because of just the nature of the products we're developing, but it's not a significant part of our organization currently.
But that's actually a really good point about the FDA. I mean, there are some very strange things happening in the industry, and new therapies coming along. The FDA have never seen them before, so they've had to give guidance where they can, but allow the biotech and pharma to move things through at risk, and to be very guarded by it. So this is actually a really exciting time.
All right, I guess everyone's ready for drinks. So thank you all for your thoughtful questions, and please join me, panelists, for the great discussion.